He's given a few talks before, and you probably heard some of this before. But I just give you a little bit of an introduction to Kevin. So, I mean, if you look at him, once he comes up here, you will see that he's very smartly dressed today in a white shirt and shiny boots and everything. And he's always very smartly dressed, but if you see him today, you can see that he's gone a long way from 3.5 years ago. So I remember when he came into my office to interview for a PhD position, one thing that I noticed was that he had really cool, tall leather boots on, and that he was fairly confident about himself, which I liked. And this has sort of come through the whole process of the PhD. So the project that he started working on was a fairly difficult one. It was a very exciting one for me and for all of us, because he got a mouse on his table, which is the RF4 knockout, and that we found doesn't mount any T-cell response. And surprisingly, that was known already for 15 years or something at that point, that these mice are pretty screwed up and can't respond in their T-cell compartment. And we had refined that a little bit more, but we really didn't understand at all what was going on, and no one did. And Kevin realized that that was an exciting project, and sort of got convinced by me that he should work on that, and took on this complicated project. And he did it with a lot of vigor and with a lot of adventurism, which is another point that I liked and still like about Kevin, because he was and is very prepared to just develop new techniques and approach different avenues that may not have been used or approached in the lab before. And what he did, and what was really important for the success of his project, was to embrace a particular area which is now probably fairly hot in immunology, which is the metabolic regulation of the immune system, which is at the level of the individual cells that take part in the immune response, to understand how not only transcription factors or cytokines that we typically look at, but also metabolic regulators contribute to the success of an immune response. And yeah, he's done that very successfully, and he could show that RF4 is a really important transcription factor for the regulation of not only classical immunological processes, but also of metabolic processes in cells. And the very recent literature that has come out from other groups in other cell types gives him right, and so supports his findings in that it now sort of comes out that RF4 is actually possibly a master regulator of metabolic function in many different cell types. And I'm sure we're going to hear more about that. So with this, up to you, Kevin.
Thanks. So good afternoon, everyone. So today I'll be presenting the work during my PhD on CD8 T-cell differentiation and its regulation by the transcription factor IRF4. So IRF4 is a member of 9 interferon regulatory factors, which have an amino-terminal DNA binding domain, recognizing interferon-stimulated response elements in target genes. Expression is mainly restricted in lymphoid and myeloid cell lineages, and unlike other interferon regulatory factors, it's not regulated by type 1 interferons, but by mitogenic signaling. And it was first characterized as a binding partner with an ETS family member, PU1, in B lymphocytes. And this is just an electrophoretic mobility shift assay with an Ig lambda light chain enhancer probe showing weak IRF4 binding alone. However, when you add a PU1, which directly interacts with the carboxyl terminal of IRF4, then you get strong binding to this sequence, indicating that it requires cooperation with other transcription factors for transcriptional activity. And IRF4 is perhaps best studied in the context of B lymphocyte differentiation, where the dosage and the transient nature or the temporal nature of IRF4 expression is critical in determining cellular fate and differentiation outcomes. So in this model, levels of IRF4 regulate mutually antagonistic gene programs that are required for either germinal-centered differentiation or subsequent germinal center exit and plasma cell development. So in this model, low and transient levels of IRF4 induce another transcription factor called BCL6, which in turn represses BLIMP1. And in this program, BCL6 is critical for the generation of germinal center phenotype B cells, which express CD95 and GL7, and also express AID required for class switch recombination and somatic hypermutation to generate high affinity antibodies. And when we have high and sustained levels of IRF4 expression, we get subsequent repression of BCL6, and BLIMP1 is switched on, which in turn induces genes required for plasma cell differentiation and high-level IgG production. And it does so by turning on XBP1 and XBP1 target genes required for high immunoglobulin production. And recently, Roger Skiamis and colleagues have identified that BCR signal strength is a critical determinant of the temporal and also the level of, or the dosage of IRF4 expression in vivo to T-dependent antigens. And this point will become a salient point later on in my presentation. So interest in IRF4 in our division came from work done by a number of our colleagues looking at the functional link between IRF4 and BLIMP1 in the context of T regulatory cell differentiation. So regulatory T cells express a transcription factor FOXP3 and are high for CD25 and are important for regulating normal immune and autoimmune responses by a number of mechanisms, including the secretion of suppressive cytokines like IL-10, and also by upregulation of inhibitory receptors such as CTLA4. And so a BLIMP reporter mouse where GFP was inserted into the BLIMP1 locus, which reports BLIMP1 transcription, were crossed to IRF4 deficient mice. And looking at affected Tregs in the peripheral, we see that IRF4 is required for BLIMP1 expression and also for T regulatory cells expressing ICOS. And this was shown to be intrinsic to an intrinsic requirement due to analysis of mixed bone marrow chimeras with an IRF4 deficient compartment, showing the loss of ICOS positive Tregs in naive mice and also following influenza infection. And so during my PhD was focused on CD8 positive cytotoxic lymphocytes, and they're an important part of the adaptive immune system that protects against intracellular viruses and bacteria. And this requires antigen recognition of peptide antigens presented on class 1 MHC molecules. And following antigen recognition, cells then undergo a process of differentiation to acquire the ability to secrete cytotoxic granules that contain granzymes and perforin, which then leads to the death of target cells via activation of the intrinsic death pathway. And CD8 differentiation entails massive clonal expansion and selection of T cells during an immune response. So in the naive repertoire, many different T cells with very different affinity TCRs for cognate antigens are present. And during the early phases of an immune response, we see this recruitment of many of different varied affinity TCR clones into the response. And this can go undergo some proliferation as well. But towards the peak of the response, only a small number of high affinity clones dominate. And low affinity cells are thought to be lost during the response or undergo early death or are preferentially diverted to become memory T cells. So we're interested in how IRF4 regulates effector and memory development. And to look at these questions, we're using several acute viral and bacterial infection models to look at these questions. And so as Axel mentioned, the phenotype of IRF4 deficient mice was published a long time ago by TACMAC's group. And this data shows a loss of cytotoxicity in the absence of IRF4 when whole splenocytes were mixed with target cells loaded with LCMV-specific peptides in a chromium release assay. So the basis of my PhD project was to uncover the mechanisms behind the lack of antigen-specific cytotoxicity, and also to further elucidate whether IRF4 was required for the acquisition of effector function, survival, or differentiation of CD8 T-cells, and also to look at the requirement of IRF4 for the development of long-term memory. And because IRF4 is critical for so many cells of the immune system, we wondered whether there was an intrinsic requirement for IRF4 and CD8 T-cell responses. And to address this question, sublethally irradiated recipients on a Li5.1 background were adaptively transferred with equal proportions of Li5.2 IRF4 deficient bone marrow or Li5.1 wild-type bone marrow, and these mice were allowed to reconstitute before intranasal influenza infection and analysis at the peak of the effector response. And looking at both spleen and lymph node in the IRF4 deficient compartment, we see that there's a complete loss of nuclear protein 366 tetramer-specific cells by class 1 tetramer staining. And in the corresponding wild-type chimeras, we can see a large proportion of antigen-specific cells. And this is just enumerated here as a proportion of the NP-positive cells of the total CD8 population in the spleen. And secretion of effector cytokines like interferon gamma and granzyme B following ex vivo peptide restimulation was also completely absent in the IRF4 deficient compartment. So we wanted to further investigate the role of IRF4 in vivo effector T-cell responses, and to do this, we utilised TCR transgenic model of the OT1 mice, which has a restricted TCR recognising the ovalbumin peptide. And so this allowed us to track the responses of a defined number of precursors early during an immune response. So in this experimental setup, we adaptively co-transferred equal numbers of wild-type Li5.1 OT1 T-cells or IRF4 deficient Li5.2 OT1 T-cells into hosts on a Li5.1, Li5.2 background before intranasal influenza overinfection and analysis at various time points during the primary immune response. And looking at 1 day post-infection in the lung-draining mediastinal lymph node, we see that there's equal recruitment of IRF4 deficient and wild-type OT1 T-cells that are primarily naive at this point. But however, looking at early proliferation by cell division di-CTV, we see that proliferation is not impaired during the early phases of an immune response in the absence of IRF4. And also other early activation markers such as CD25 and CD69 show normal upregulation. So this just shows the level of expansion of IRF4 deficient and wild-type OT1 T cells in the spleen and lung at day 3 post-infection. And at this point, it looks relatively normal. However, when we look later on during the immune response, at day 5 and day 10 post-infection, we can see that wild-type T cells massively outcompete IRF4 deficient OT1 T cells. And this is just quantified here as a ratio of knockout versus wild-type OT1 T cells in the spleen and lung over time. And looking at effector function following ex vivo peptide restimulation, we see that IRF4 deficient T cells are able to produce some effector cytokines like interferon gamma and TNF alpha, and also granzyme B at relatively normal levels early on during an immune response. However, looking just 2 days later, we note that the majority of the granzyme B positive T cells are lost, and also cells double positive for interferon gamma and TNF alpha seem to be reduced. So this suggests to us that IRF4 is critical for maintaining effector function and survival of activated effector T cells. And so just to summarize this first part, the data I've shown you demonstrates that in the absence of IRF4, we get normal proliferation in vitro and in vivo, and there's a lack of accumulation of activated cells and a progressive loss of antigen-specific cells over time during an immune response. And this really phenocopied low affinity TCR stimulation in wild-type T cells. And in a study by Zen and Bevan, where they utilised altered affinity over ligands expressed on recombinant listeria to look at the selection and expansion of wild-type OT1 T cells in the peripheral, we note that they found that the highest affinity ligands for the TCR induce the greatest amount of effector T-cell expansion in a graded effect, in a graded way. And the lowest affinity over ligands induce the lowest levels of T-cell expansion. And because we saw many phenotypic characteristics that were similar to low affinity stimulated T-cells, we wondered whether there was a relationship between TCR affinity and induction of IRF4 expression. And so to look at this question, we utilize the altered affinity over ligands, the native N4, intermediate Q4 and low affinity V4 ligands, either as peptide or as is expressed on recombinant listeria. And so in vitro activation of wild-type OT1 T cells with IL-2 and the altered affinity over ligands demonstrated to us that induction of IRF4 expression was dependent on TCR affinity, with the highest affinity ligands inducing the highest amounts of IRF4 protein, more so than the intermediate or low affinity ligands. And so we wanted to test also if this regulation occurred in vivo, and to do this we adoptively transferred Li5.2 marked OT1 T cells into naive Li5.1 recipients and infected them with the altered affinity Listeria ovarians. And by intracellular IRF4 staining, we also observed that IRF4 protein expression correlated with the level of affinity of the ligands for the TCR, with the highest affinity N4 ligands inducing the highest levels of IRF4 protein. So we also wanted to know or test if the dosage of IRF4 expression or the levels had any effect on T-cell expansion or in vivo immune responses. And so to address this question, we co-transferred wild-type OT1 T cells alongside Li5.2 marked heterozygote or IRF4 deficient OT1 T cells into naive hosts before infection with Listeria expressing high or low affinity ligands. And in all situations, wild-type T cells expressing normal levels of IRF4 outcompeted heterozygote and deficient OT1 T cells under conditions of low and high affinity stimulation. And in the absence of IRF4, high affinity ligands failed to induce any increase in clonal expansion, while in heterozygote and wild-type cells, we saw that the level of T-cell expansion increased with TCR affinity. And this was also confirmed by CTV analysis of transferred cells, where at day 2 post-infection, we can observe a high, IRF4 high and low population on CD44 positive activated cells. And looking just 1 day later, we observed that it was the IRF4 high population that was preferentially underwent expansion in vivo, suggesting that dosage of IRF4 was critically important for qualitatively for and quantitatively for effector T-cell responses in vivo. So the data I've just shown you demonstrates that IRF4 is essential for CD8 T-cell responses and is regulated by TCR affinity. And our next hypothesis was that IRF4 functions to translate TCR affinity signals into the appropriate transcriptional program regulating effector development. And we used 2 approaches to address this question. And the first was to conduct RNA sequencing of wild-type and IRF4 deficient OT1 T cells with high and low affinity peptide stimulation. And in a second approach, we conducted chromatin immunoprecipitation and sequencing to identify genes directly bound by IRF4. And RNA sequencing of low and high affinity stimulated wild-type cells demonstrated that IRF4 is required for genes that are induced depending on TCR affinity. And this is just a heat map showing the 250 most differentially regulated genes between high and low affinity stimulated wild-type cells. And as you can see, high affinity TCR stimulation is required for the transcriptional repression of some genes here, and also for the transcriptional upregulation of some genes as shown here. And when we look at the same 250 genes in the absence of IRF4, comparing low and high affinity TCR stimulation, we note that the direction of expressional changes is lost, and that the IRF4 deficient cells have more in common with a low affinity stimulated wild-type T cell than it does with a high affinity stimulated wild-type T cell. And this is also demonstrated by global transcriptional analysis shown here in a Pearson correlation analysis of global log-fold changes in genes differing in affinity, and also genes that are differing between wild-type and knockout T cells. And we can see that the correlation of expression of these genes is very high. And when we conduct the same analysis in the absence of IRF4, differing between affinity in IRF4 deficient T cells, and also compare them to the expression of genes that differ between knockout and wild-type T cells, we see that this correlation is lost, indicating again that IRF4 is required for genes that are induced depending upon TCR affinity. So we also wanted to dissect whether any of these genes that were differentially regulated were directly regulated by IRF4. So we conducted chromatin immunoprecipitation with anti-IRF4 antibodies, and then deep sequenced these samples. And this is just an example of one target gene of IRF4, PIDM1, which encodes BLIMP1, an important transcription factor for terminal affected differentiation. And in our analysis, we uncovered 2,268 significant peaks, of which 629 showed differential transcript expression. And de novo motif analysis of the most significant binding peaks revealed that the majority of IRF4 binding sites coincided with a composite IRF4 AP1 binding sequence, as shown here in these different motifs. And this was also conserved and also shown recently by several groups to be a conserved mode of binding in CD4 T cells and dendritic cells. So network analysis of our RNA sequencing data between genes that were differentially expressed between IRF4 deficient and wild-type T cells by Ingenuity's pathway analysis revealed that the most differentially expressed gene network was one involved in carbohydrate metabolism, molecular transport, and small molecule biochemistry. So this type of analysis collates experimentally observed and direct and indirect interactions between gene targets from experimentally observed data from curated literature sources. And fitting with this, we saw that two transcriptional regulators of T-cell metabolism were direct IRF4 targets, and one of them was FOXO1, which is an important regulator of T-cell quiescence and migration, and the other being HIF1 alpha, hypoxia-inducible 1 alpha, which is a transcription factor that is involved in regulating glucose and pyruvate metabolism. And this just demonstrates here that transcript expression of those two factors were downregulated, along with related factor FOXO3. Naive and memory T cells have a metabolic program that is very distinct from activated effector T cells, and they're considered metabolically quiescent and primarily utilize mitochondrial metabolism, known as oxidative phosphorylation and also fatty acid oxidation. This process is efficient at producing ATP, but also requires high oxygen levels. Upon antigen recognition, effector T cells undergo a reprogramming of mitochondrial metabolism to utilize cytosolic aerobic glycolysis and also glutaminolysis. Using these pathways produces low amounts of ATP, however, yields metabolic intermediates that are essential for de novo synthesis of nucleotides, amino acids, and lipids required to produce a new cell during massive clonal expansion. Because we saw that genes that were enriched for carbohydrate metabolism were regulated by IRF4, we decided to look at the components of the glycolytic pathway. And here just shows data from our RNA sequencing experiments with a number of rate-limiting components of the of the glycolytic pathway being downregulated in the absence of IRF4. And these included glucose transporters, GLUT1 and GLUT3, phosphofructose kinases, which catalyzes an irreversible step in glycolysis, and also aldolases and pyruvate dehydrogenase kinase, which is important for deciding whether pyruvate enters the mitochondria for OXPHOS or whether it becomes processed in the cytosol. And some of these were also direct gene targets of IRF4, and these included hexokinase 2, catalyzing the first step of glycolysis, GLUT3, and glycogen phosphorylase. And some of these genes were also confirmed by qPCR analysis at earlier time points following anti-CD3 activation in vitro. And the white bars indicate the transcript levels of these genes in IRF4 deficient T cells. And as we can see, following activation, expression of a number of components of the glycolytic pathway is downregulated in the absence of IRF4. And an earlier study by Douglas Green and colleagues also identified that an early transcription factor, c-MYC, also controls metabolic programming through upregulation of gene networks involved in glycolysis and glutaminolysis. However, we found that IRF4 was dispensable for c-MYC expression following activation, and so suggesting that both IRF4 and c-MYC act in concert to regulate expression of these genes. Because we saw a loss of expression of many of the important components of glycolysis, we wanted to look at whether IRF4 deficiency resulted in impaired glycolysis in vitro. And firstly, we looked at facilitated glucose transport using a fluorescently labeled glucose analog 2-NDBG, comparing naive rested T cells in IL-7 with T cells that were stimulated with anti-CD3 for 24 and 48 hours. And as you can see in the wild-type T cells, compared to the naive resting T cells, glucose transport is increased upon T cell activation. And this process is impaired in the absence of IRF4. We also measured glycolytic activity by real-time extracellular metabolic flux assays. And in this assay, we measure extracellular acidification rate, which is an indicator of glycolytic activity. And lactate produced by the cell produces a proton which leads to acidification of media, and this is a surrogate marker for glycolytic flux. And on the left here is the basal glycolytic rate of wild-type and IRF4 deficient T cells following anti-CD3 stimulation. And we can see in the absence of IRF4, upregulation of glycolysis is impaired. And this is also shown here over time. So T-cell activation also requires intact mitochondrial function and electron transport chain function. And the functional link between mitochondrial function and TCR activation is believed to be from the increased intracellular calcium flux that results from TCR stimulation, which is then taken up by the mitochondria to produce ATP and NADPH, which is subsequently used for aerobic glycolysis. And a study by Erica Pierce and colleagues have identified using chemical inhibitors to complex 5, also known as ATP synthase, that shutting off ATP synthesis generated during OXPHOS impairs T-cell proliferation and early activation. And just some other inhibitors I will mention during the next few slides. FCCP, which is a protoionate 4, which uncouples proton pump to ATP synthesis. And also antimyosin A and rotenine, which inhibits complex 3 and 1 of the electron transport chain, respectively. We also decided to look at mitochondrial function as in addition to aerobic glycolysis, because pyruvate produced during glycolysis can feed into the mitochondrial tricarboxylic acid cycle and affect mitochondrial metabolism. First off, we decided to look at mitochondrial membrane potential as a surrogate marker for oxidative phosphorylation activity. And here we're just comparing naive T cells with activated wild-type and IRF4 deficient T cells. And this mitotracker dye is sequestered in respiring cells and is retained depending on intact mitochondrial membrane potential. And as you can observe, at 24 hours after activation, IRF4 deficient T cells have increased mitochondrial membrane potential. However, this is rapidly decreased over time relative to wild-type T cells. And we also confirmed this by using extracellular metabolic flux assays, looking at oxygen consumption rate, which is an indicator of OXPHOS. And as you can observe here, we only observe mitochondrial defects at 48 hours following activation, while we observe earlier defects in glycolysis. And this is also reflected here in the decrease in ATP synthesis following the addition of ATP synthase inhibitor oligomycin. So we also just wanted to confirm whether glycolysis was impaired in vivo. And to look at this question, we utilised the Listeria overstrains and infected naive recipient mice before adoptive transfer of wild-type or IRF4 deficient Li5.2 marked OT1 T cells, and sorted affected T cells that were activated at day 4 post-infection before subjecting these cells to a glycolytic stress analysis. So at the beginning of the assay, these cells arrested at glucose and pyruvate replete meeting that this acidification rate seen here is due to non-glycolytic pathways. And over time, glucose is added into the media to stimulate glycolysis before addition of oligomycin, which induces maximal glycolytic capacity of cells. And here we can see that following the addition of glucose, IRF4 deficient T cells failed to respond to the same level as wild-type T cells and have no glycolytic reserve. Because we also observed a strict dependence of IRF4 for TCR affinity and strength signals, we wondered whether there was a link between IRF4 expression and also conversion to aerobic glycolysis in vivo. So we utilised the altered affinity Listeria overstrains to look at induction of aerobic glycolysis in vivo following adoptive transfer of wild-type and IRF4 deficient T cells. And here just is data showing facilitated glucose transport in response to high and low affinity ligands. And you can see that with high affinity ligands, we get a much greater induction of glucose transport into the cells compared to low affinity ligands. And in all situations, regardless of the strength of TCR signaling, IRF4 deficient T cells resemble low affinity stimulated T cells in their glucose uptake. And this was also confirmed by assaying extracellular lactate production, which is the end product of glycolysis. And as we can see here, the high affinity ligands induce most extracellular lactate production, while low affinity ligands are similar again to the IRF4 deficient T cells. And this is also reflected by impaired blastogenesis of IRF4 deficient T cells in vivo to high affinity TCR ligands. So this is just a measure of cellular size forward scatter. And as you can see here, that wild-type T cells increase in size depending on the strength of the TCR affinity, while in all situations, IRF4 deficient T cells remain a lot smaller. And this was also reflected in the loss of several receptors that are important for metabolic function, and one of these is the transferrin receptor CD71, which also, again, is lost in the absence of IRF4. So this demonstrated to us that IRF4 linked TCR affinity with the induction of aerobic glycolysis in a graded manner. Which brings us back to our model of clonal selection and competition of T cells during a primary immune response. So in the naive repertoire contains clonotypes of many different varied TCR affinity, and these are all recruited into the primary immune response at some level. But T cells with low affinity TCRs tend to go earlier death and aborted expansion. And at the peak of the response, it is dominated by a few high affinity TCR clones. So there is a focusing of the repertoire. So the mechanism for this process was previously unrecognized, and what we've demonstrated here is that TCR affinity induces a graded level of IRF4, and it's the cells that express the highest levels of IRF4 that undergo the greatest expansion due to their increased ability to metabolize glucose and utilize aerobic glycolysis for effector function and expansion. So just to truly test whether levels of IRF4 were a limiting factor during in vivo low affinity stimulation, we activated wild-type OT1 T cells and transduced them with a retrovirus containing an IRF4 overexpressing GFP virus or a GFP control virus, and rested these cells in IL-15 for 3 to 4 days before adaptive transfer into recipients that then received a low affinity ligand in vivo. And as you can see here, cells that overexpress IRF4 underwent a greater level of expansion than the GFP control to low affinity ligands in vivo, demonstrating that IRF4 was a limiting factor in situations of low affinity stimulation. So what I've just shown you is that IRF4 is essential for a productive CD8 T-cell response, and induction of IRF4 is TCR affinity dependent, and it also functions to translate TCR affinity-driven signals required for metabolic reprogramming and clonal expansion of CD8 T cells. So next, we wanted to know whether there was a requirement for IRF4 in the development of long-term immunological memory. And I guess an outstanding question was whether memory T cells express IRF4, or whether downregulation of IRF4 was important following the clearance of antigen, perhaps to allow for the upregulation of oxidative phosphorylation and fatty acid oxidation that are critically important for long-term survival of memory cells. And another outstanding question was whether there are different levels of IRF4 in different memory T-cell populations, and whether this dictated the different phenotype of memory cells and the different different tissue localization, such as that of resident memory T cells compared to circulating central memory T cells. So we just wanted to confirm whether IRF4 was expressed in memory T cells. And we looked in vivo first using influenza overinfection, where we adoptively transferred Li5.2 marked wild-type OT1 T cells into Li5.1 recipients, and then looked at IRF4 expression by intracellular staining on CD44 positive activated T cells. And as you can see, IRF4 expression peaks early on during the effector immune response and is subsequently downregulated, except at memory time points, they have a basal level of expression that is above that of naive T cells. And this was also confirmed by qPCR analysis using another infection model, LCMV, where sorted GP33 positive antigen-specific cells from day 10 and day 30 show that IRF4 transcript is downregulated in memory development. And we also wanted to see the response of IRF4 expression to memory-conducive cytokines such as IL-15. And when we compare stimulation of T cells in IL-2 to IL-15, we note that both transcript and protein expression of IRF4 is downregulated in response to IL-15. And because IRF4 was expressed at such a low level in memory T cells, we wondered whether there was really a requirement for IRF4 in memory T cell responses. And so we utilized conditional IRF4 floxed mouse, where Cre recombinase was placed under the promoter elements of the granzyme B gene, and this was also coupled to a GFP reporter. So cells expressing GFP have also undergone a deletion of IRF4. And so we sorted GFP, GP33 positive cells from IRF4 heterozygote and deficient mice before adoptively transferring them into naive recipients that were allowed to rest to a memory time point to either look at the maintenance of memory cells, or were subsequently reinfected with LCMV to look at the recall response. And as you can see here, in uninfected mice, just looking at the maintenance of memory cells, we observe that there's no defect in the absence of IRF4 for the maintenance of memory cells when we compare the heterozygote and IRF4 deficient generated cells. But however, following secondary reinfection, we note that it's only the cells that express IRF4 that undergo secondary re-expansion, while the IRF4 deficient cells are lost during this response. Because we saw that IRF4 was dispensable for homeostatic proliferation in vivo and maintenance, we wanted to further characterize whether that was truly the case. And so to look at this question, we generated in vivo memory cells by either adoptively transferring wild-type OT1 T cells or IRF4 deficient OT1 T cells into mice that were infected with Listeria ova, and I re-isolated CD44 positive T cells at day 42 post-infection before CTV labeling these cells and adoptively transferring them into mice, and then analyzing them at day 30 to look at homeostatic turnover. And to our surprise, in the absence of IRF4, these cells underwent a lot greater proliferation in vivo under homeostatic conditions. And this could be partially explained by the loss of cytokine receptors for homeostatic cytokines such as IL-15 and IL-7. And here we can see in red that in the absence of IRF4, we have a lower expression of IL-7 receptor alpha and also CD122. So I've shown you that IRF4 is essential for memory T cell function, but is expressed at low levels in memory T cells and is required for proper homeostasis and secondary recall capacity of memory T cells. And part of this work is ongoing to elucidate how IRF4 is truly important for regulating memory T cell function. But another question we had was whether IRF4 also played a role in the transcription regulation of the exhaustion program during chronic infection. And in chronic infections differ from an acute infection in that there's persistence of antigen that can remain in the host for up to several years. And when there's persistent antigen, we get altered effector differentiation, and also massive clonal deletion to prevent overwhelming pathology. And this program is sometimes referred to as exhaustion of T cells, and it's characterized by the loss of interferon gamma, TNF alpha, and IL-2, respectively, and also upregulation of inhibitory receptors such as PD-1. And also, these cells express higher levels of the T-box transcription factor EMS. These cells also differ from memory T cells in that they express lower levels of IL-7 receptor and CD122 and require antigen for their maintenance, unlike memory T cells. So we were just interested to know whether there was a link between an increase in or persistence of antigen IRF4 expression and whether this meant that exhausted T cells also had an altered metabolic state. And to look at this question, we compared wild-type P14 transgenic T cells with a TCR specific for the GP33 peptide of LCMV, comparing acute LCMV infection with a chronic LCMV strain. And as I mentioned before, chronically infected antigen-specific cells express high levels of PD-1 and do not express lower levels of IL-7 receptor. So first, we just wanted to look at glycolysis by glucose uptake by 2-NBDG staining. And as we can observe here, the PD-1 positive cells take up more glucose than cells derived from an acute infection. And also mitotracker staining was also increased in the PD-1 positive cells acquired from the chronically infected mice, suggesting that T cells or exhausted T cells display higher basal levels of metabolic function than acutely infected T cells at an effective time point. And we also just wanted to confirm this by real-time extracellular flux analysis and looking at the basal ECAR rate, which is an indicator of glycolysis. We note that the cells isolated from chronically infected mice had a higher basal level of glycolysis than the acutely infected T cells. However, when we re-stimulate cells with PMA and onomycin during the assay, we note that it was only the cells derived from an acute infection that could re-elaborate aerobic glycolysis to a maximal extent, while this was impaired in chronically infected T cells. And this was also demonstrated ex vivo by reculturing these cells in IL-2 to stimulate glycolysis. We did this to measure extracellular lactate production. And as you can see, in cells that have an exhausted phenotype, we're less able to recapitulate glycolysis. And we also looked at mitochondrial function in chronically infected cells compared to cells derived from an acute infection. And as we can, and looked at a measure called mitochondrial spare respiratory capacity. And mitochondrial spare respiratory capacity is the capacity of the mitochondria to produce energy under cellular stress and also conditions of low nutrient availability. And this is believed to be critically important for the long-term survival of memory T cells. And mechanistically, it's believed to be upregulated by memory-promoting cytokines such as IL-15, and also via the upregulation of genes involved in fatty acid oxidation. So spare respiratory capacity is calculated as a difference between the basal rate of oxygen consumption and that following the addition of oligomycin and FCCP here. And as you can see, the cells derived from a chronic infection that have high levels of PD-1 are lower in their spare respiratory capacity compared to acutely infected cells. Because we saw these metabolic alterations, we wanted to also confirm that these multiple metabolic parameters were associated with an increase in IRF4 expression or whether it was due to some other mechanism. And previously, John Werry's group has published that chronically infected cells that express PD-1 were also high for the transcription factor EMS. And we just wanted to assess IRF4 levels in vivo by intracellular staining by comparing acute and chronic infection. As we can see here, during an acute infection, most of the cells are PD-1 low and IRF4 low. And the same P14 T cells from a chronic infection showed increase in PD-1 and also an increase in IRF4 protein expression. And this was just confirmed here by qPCR analysis of sorted GP33 positive cells from acute and chronic infection, showing an increase in IRF4 message. Because we saw an increase in IRF4 expression and also all these metabolic differences between acute and chronic T cells, we wanted to know whether changing the dosage of IRF4 expression would change the outcome of or the phenotype of exhausted T cells. And to do this, we compared wild-type P14 T cells with P14 T cells lacking one copy of IRF4 and looked at several things, including inhibitory receptor expression, glucose uptake, and mitochondrial membrane potential, comparing acute and chronic LCMV infections. And when we take away one copy of IRF4, we note that several of these parameters that define exhausted T cells are reverted back to seemingly normal levels. They are more comparable to the acutely infected T cells than they are to chronically infected T cells. And this was reflected by the lower expression of inhibitory receptor PD-1 and also lower glucose uptake, indicating that the increase in glycolysis does not occur, and also a restoration of normal mitochondrial membrane potential when we have heterozygote levels of IRF4. So this indicated to us that the levels of IRF4 affect the magnitude of glycolytic metabolism exhibited by T cells, and that IRF4 is a dose-dependent regulator of metabolic reprogramming of T cells during acute and chronic infection. And so this is just some of the current work I'm doing in the lab. And I guess currently I'm aiming to further characterize the phenotype of IRF4 deficient and BATF deficient T cells during a chronic infection by using several techniques like RNA sequencing and overexpression of IRF4 to see if that also recapitulates some of the phenotypes of chronic infection. And so this leads me to thank everyone in Axel Kelly's lab, and in particular Axel for his support over the years of my PhD, and also some other members of the lab that contributed data to this project. And one of them was Mia and Annie, who helped with chip experiments. And also members of my PhD panel, Phil, Gabrielle and Steve. And also like to thank members of the Infection and Immunity for their help with the LCMV experiments, and all the animal technicians. And much of the bioinformatic analysis was done by WEHI and Gordon Smythe. And also we have collaborators at the Baker IDI who help with the metabolic assays, and that's in the lab of Mark Fabre. And so lastly, I'd just like to thank family and friends who've supported me over the years. And yeah, thanks for attending today. Thanks.
Well, I think that was a great presentation, and I'm sure there's some questions. Someone?
One of the graphs of the or knockout, there was CBIC going up a bit.
Yeah, I mean, it looked like there was increased c-MYC protein expression.
Is that potential for the cells to drive integration faster?
It might be a compensatory mechanism, but I guess we won't really know that until, only if we have like c-MYC mice to cross them to. But maybe they're more dependent on c-MYC in the absence of RF4 to induce expression of all these genes following activation. It's one possibility.
I noticed that you made one point, I'm not sure whether you meant to make that, in your increased homeostatic turnover of the memory cells in the absence of RF4, you said one explanation could be that they express less ISM receptor and less I-15 receptor. Didn't quite get the reasoning for that. Wouldn't you expect them to turn over less if they have less of the receptor for the homeostatically important cytokines?
Well, I thought they would probably respond more like effector T cells in that situation. So I'm not really sure how that really fits with the data I'm seeing. But one explanation was that RF4 deficient T cells also express lower levels of FOXO factors, which are known to be important for regulating quiescence of at least naive T cells, but not much is known if they also regulate the same processes in memory T cells. So that was one kind of link that we had there. And I've done a few by Western blot and qPCR analysis that FOXO1 levels are, the export of FOXO1 is increased in the absence of RF4 from the nucleus. So that was one possibility. But I guess that's ongoing work, so.
So you had, in your ChIP-seq analysis, you had about 2,800 peaks, and of those 600 were associated with differentially expressed genes.
Yeah.
The remaining 2,200 peaks, were they associated with genes, or were they?
So there was 2,260 or so significant binding peaks. So the other 1,600 could have, like the differential expression was too low for the cutoff that we had. But many more were differentially expressed, but just not significant. And
so were the other peaks, were they associated with genes, or were there also peaks in?
Well, the majority of the peaks that we found that were differentially expressed were intronic or in promoter regions, but I'm not really sure about the ones that weren't changed.
I think we tried to look at that at that point quite carefully, that there was definitely many peaks outside of actual gene bodies, but I don't think we found a very clear association between positioning of the peak and the actual outcome in transcription, but that could be analyzed differently, I guess.
Can you speculate what is the sort of the mechanism that translates the T-cell affinity, like high and low, to the high and low expression of the IRF?
Well, NF-kappa B family members are important, and we have some data suggesting that CREL is important for inducing protein expression of IRF4. And also another group, Leslie Berg's group, had found that ITK knockouts also don't express IRF4. So they found that that was a critical TCR transducing molecule to induce expression of IRF4, but there could be many other pathways.
So the experiments where you put in IRF4 with the retrovirus and your low affinity activated cells came up in terms of number. And a parallel question is that IRF4 seems to be specifically expressed in some cell types and not others, yet many different cell types need to undergo a proliferative burst, both in the immune system but also in other cells. If you were to put an IRF4 into other cell types with a low mitogenic stimulus, would they also proliferate? Is it only specific to T cells? What about other immune cells?
I haven't looked at other immune cells, but I'm guessing the mechanism is conserved at least for B lymphocytes. But yeah, I mean, that remains to be seen whether it's the same for other or non-immune cells or...
There is no other questions. Would you like to thank Kevin?